Hello there, I'm Christina, Food Technology Research Scholar from MIT ADT University, Pune. And today we are going to learn about the various applications of food science and technology in the field of molecular gastronomy. These are the contents for today's slide, and by the end of this presentation, you will learn that how simplest food recipes and ingredients can be made so fascinating and take the dish to the next level, yet keeping the traditional uniqueness of those recipes. So, what is molecular gastronomy? So, what do you come across when you first heard of this term? Well, the term gastronomy is taken from the Greek word gaster, meaning stomach, and nomia, meaning law. Molecular gastronomy is a scientific discipline concerned with the physical and chemical transformations that occur during cooking, or in other words, the chemistry that is relied on to create unexpected dishes. Next is what is the importance and why do we need this field? So it includes exploring existing recipes, introducing new tools, ingredients, and methods into the kitchen, also to help general public understand the contribution of science to the society, also to develop novel textures, stabilizing formulations, and improve overall product quality. And this can be achieved by collaboration among chefs and scientists. Chefs are the experts in cooking, and scientists know the chemistry and interaction between food molecules during cooking. So, the knowledge and experience of both chefs and scientists can be used in this field. So, when they both come together, food recipes and dishes can be taken to next level by improving texture, formulation. So these are some of the ways how we can how the molecular gastronomy can be useful. First is by implementing new techniques, ingredients that aren't usually used in the kitchen. We will be going to study about these ingredients in the next slides. Next is food pairing. It is one of the major challenges chefs face. Different food ingredients can be paired using molecular gastronomy and combination of dishes can be tested and ultimately it will enhance the sensory and nutritional properties of the food. Also, it addresses the various kitchen challenges that are usually faced by identifying the right time temperature combination to preserve the nutrition of the food and also to maintain the color of the vegetables during cooking. Usually the green color of the vegetables is lost, so that is due to the loss of magnesium ions from the chlorophyll. So instead of that, we can incorporate copper ions during cooking which will maintain the green color of vegetables. So these are the key ingredients. First is agar-agar that is used for creating gel-like structure. Next is calcium lactate that is used in the surface molecules or like it is used as a membrane to hold the liquid content intact. Next is xanthan gum, sodium alginate, soy lecithin, it acts as an emulsifier. Next is liquid nitrogen, maltodextrin, and transglutaminase. So these are the techniques that are used in molecular gastronomy. First is jellification. As the term itself denotes, it is the transformation of liquids into gels using agar-agar pectin. So basically it improves the overall mouth feel of the particular food product. Next is purification. It refers to the process of adding sodium alginate to the liquid and transforming into spheres that resemble caviar or tapioca pearls. So caviar is basically a product from the fish. Next is emulsification. We all are familiar with this term, like it is basically mixing of two immiscible liquids using emulsifiers, like we studied in the ingredients that is soy lecithin. Next is siphon whipping. In this, the device siphon is used and it uses a pressurized gas to create foams and whipped creams, and that is used in culinary arts as toppings, infusing fruit in cold coffee, etc. Further, we have deep freezing. We all know deep freezing basically views liquid nitrogen and it is used to freeze the ingredients. Next in line is meat gluing. This technique is very interesting. There is an enzyme called transglutaminase that can speed the protein reactions between the substances, and it is often used as an additive in meatballs and bacon meat, and it can be given new shapes. Next is deconstruction, and this the traditional dishes or recipes. From that, the ingredients are isolated, and that itself is created into a whole new recipe. Next is converting liquid into powder. So in this special chemical that is obtained from maize, that is N-sorbate, so any liquid can be directly converted into powder. These are the examples of molecular gastronomy. First, we have honey caviar with forme d'ambre and black tea. So here we can see the caviar-shaped pearls where the honey is intact within the calcium chloride and alginate membrane, and it uses the reactions to trap liquid ingredients within this extremely thin surface. Next, we have peanut butter powder and papaya agar-agar noodles. Interesting, no? The gelling agent is mixed with liquid ingredient and brought to a boil, then later passed through a clear silicone tube under an ice bath to get perfectly shaped gel noodles. So this is an example of the enzyme, how it can be used, that is transglutaminase. So the enzyme is basically applied on the surfaces of the meat, and both of the meat can be glued together to give the desired shape. These are the tools that are used. These are not very high-end tools, but these are developed according to the convenience of the use and they are named accordingly. First is anti-griddle, smoking gun, ultrasonic probes, siphons, hypodermic syringe, vacuum machine. So yeah, next we have I have given you some of the examples like hotels and restaurants where you can experience these molecular gastronomy foods. Of course, the prices are a little high, but if you want to treat yourself, so definitely you can try it. First is Masala Library that is situated in Mumbai. Next is Molecule Air Bar that is in Gurugram. Next is Capability in Bangalore, Fuzzy Cafe in Pune. So yeah, we have come to the end of this slide. No doubt the field continues to evolve and it not only enhances the sensory experience of dining but also provides deeper understanding of food chemistry. And of course, yes, as technology advances, possibility will emerge. These are the references I have taken. You can definitely refer for your deeper understanding of this topic. Hope you enjoyed learning and thank you.